Hello, and welcome to my presentation of zinc homeostasis in land plants. Now, today we'll mainly be focusing on Arabidopsis studies, but it should be noted that the tolerance to zinc usage efficiency actually varies a lot within a species and between species. However, a lot of the principles here can be widely applicable to other plants. So, why should we care about zinc homeostasis in land plants? Well, zinc deficiency actually leads to a large reduction in crop yields. Zinc deficiency in humans actually leads to a large number of deaths on an annual basis, and an increased amount of our land is contaminated with high levels of zinc. So we really need to know how do plants respond to high and low levels of zinc, and how do they allocate zinc within the plant. So, zinc, what is it actually good for? Well, quite a lot actually. 10% of the eukaryotic proteome binds zinc with a variety of different ligands and geometries. Now, binding of zinc can be used for catalysis. For example, in some superoxide dismutases have copper and zinc at the active site. Now, superoxide dismutases convert the highly reactive oxygen species superoxide to a less reactive oxygen species hydrogen peroxide. So this is useful in ROS detoxification. And also, zinc is used as a structural role in some zinc finger transcription factors. This is significant because zinc is redox inactive. It only has the +2 form in comparison to iron, which has +2 and +3. And if you kind of substitute zinc for iron there, you get an increased level of reactive species being generated in quite close proximity to DNA. So that's why zinc is probably more safe than iron for transcription factors. If we want to understand zinc in plants, we also have to understand where a plant gets their zinc from. Now, this is from the soil, obviously, and zinc in the soil can be simplified into being in 4 phases. Now, there is zinc ions that are free and liquid zinc ligand species. These are both often available to plants. And then there are the unavailable phases of zinc, which is the adsorbed exchangeable zinc ions and the solid phase zinc. And there's movement between these different pools, as you can see. There are several different factors which can reduce zinc availability to plants, and this includes high pH. Now, high pH will increase adsorption of zinc to anionic soil particles. Some organic matter can chelate zinc into the solid phase. There are competing metals which will compete with zinc for binding sites on, for example, plant roots. Reducing conditions can favor the production of insoluble zinc sulfide, which is unavailable. And sandy soils which have very limited cationic binding capabilities, and so a lot of zinc is leached from these soils. Now, zinc deficiency is a global issue, as you can see from this map, with grey being medium zinc deficiency and black being widespread zinc deficiency. This low zinc availability in soil then leads to zinc deficiency symptoms in plants, including crops. These symptoms are chlorosis, growth stunting, and infertility. Now, humans which rely on these crops for a large percentage of their calories can also get zinc deficiency, and this has quite serious symptoms, including skin issues, growth stunting, and reduced immunity. It's currently thought that around 800,000 deaths of under 5s every single year result from zinc deficiency. So this is quite a serious issue. Not only are there areas of very low zinc, but also regions of very high zinc, often near industrial activity. Now, in non-adapted plants, this can cause symptoms of chlorosis, stunted growth, and necrosis. However, there are a few species that have adapted to live under these conditions. Now, one type of this plant is a hyperaccumulator plant. Now, as the name suggests, these have increased sequestration of the metals, often in storage organelles or storage organs, and this allows them to adapt to these environments. These plants are actually quite interesting for both phytoremediation efforts, but also phytomining possibilities. Now looking towards plant zinc uptake, 2 general strategies are employed. In strategy 1, acidic compounds and low molecular weight ligands like the citrate shown here are exuded by the roots. This increases zinc solubility and therefore aids zinc uptake. In strategy 2, which is only used by cereal plants, phytosiderophores can be exuded like this Smaczniak acid shown here. Sorry if I butchered the pronunciation. This is a kind of funky non-proteinaceous amino acid that's exuded into the soil again. These bind zinc with quite high affinity, and then the complexed form with zinc can then be uptaken by the roots. So if we go from the soil now into the plant cell, so we're looking at cytosolic zinc buffering and muffling. First of all, I want to look at the levels of zinc in the cell, which is on the left there, scaling from low zinc to high zinc. Now, when you have excessive amounts of free zinc ions, the zinc outcompetes other metals like iron and copper, and this forms mismethylation and metal deficiencies. Then you have the optimal range, which has been seen to be around kind of the picomolar range in different organisms. And then without sufficient free zinc, you have reduced membrane stability and reduced activity of zinc-containing enzymes, like the superoxide dismutase I was mentioning earlier. Now, in steady-state conditions, the level of free zinc ion in the cells is controlled by buffering. This is where you have an equilibrium established between zinc-binding ligands, such as zinc-binding proteins, and the free zinc in solution. Then there is non-steady state conditions. This is where, for example, you have a sudden large increase in zinc for whatever reason. Now here, you have to have activities of both the zinc-binding ligands, but also the activities of various transporters that can then reduce the amplitude of the sudden rise in zinc in this example. The cytosolic zinc binders in the cell fall into two categories, high molecular weight ligands and low molecular weight ligands. Firstly, I want to tackle the high molecular weight ligands. This could be metallothioneins and phytochelatins. Now, the metallothioneins are cysteine-rich small proteins that bind transition metals, not just zinc here, and the phytochelatins are small peptides synthesized by phytochelatin synthetase from small units of glutathione. Now, the structure of these phytochelatins is shown below, and you can see it's actually made of a gamma peptide bond between glutamate and cysteine, and then there's also a terminal glycine in the repeating unit there. Onto the low molecular weight ligands, you've got nicotianamine shown below and histidine. Now, nicotianamine is actually another non-proteinaceous amino acid synthesized from the condensation of 3 SAM molecules or S-adenosyl-methionine molecules by nicotianamine synthase. And there's also histidine that forms a less important role in zinc cytosolic buffering. Subcellular zinc stores are also very important in zinc homeostasis in plants. For example, the vacuole is very important for storing zinc under excess conditions. Many mutants in vacuolar transporters that transport zinc into the vacuole show reduced fitness in high zinc levels, backing up that claim. Now, moving on to the endoplasmic reticulum, this is where secretory proteins are folded, and disruption in this folding can be signaled through the unfolded protein response, or the UPR. Now, it's been shown that zinc levels in the ER are actually controlled to initiate this UPR under salt stress conditions. And my final point here is symplastic pathway, where zinc in the ER could be one of the ways in which zinc symplastically moves between cells towards the xylem. I'm going to start talking about some of the specifics involved in zinc homeostasis. Firstly, the transporter families. Now, I've listed 6 here. There are a bunch more, and it would take me all day to go through the specifics of each specific protein in each family, so I'm not going to do that. What I'm going to do is I'm going to talk about the general features of transporters and what I think are the important features of transporters worthy of study. So there's the transport substrate, i.e., is it transporting just zinc by itself? Is it transporting zinc complexed with nicotianamine, like some of the YSL proteins? Now, moving on to transport selectivity, is it just transporting zinc, or is it transporting iron and magnesium or manganese? IRT1 is a very popular zip in Arabidopsis, and it transports not just iron, but also zinc and also manganese. So when you upregulate it to try and increase iron uptake, you're also going to increase the uptake of zinc and manganese. And obviously this has biological consequences. ATP hydrolysis, where you need that hydrolysis to pump against a concentration gradient. Direction of transport. Now, to simplify this, zips normally transport into the cytosol, and these other proteins normally transport zinc out of the cytosol. Localization. Obviously, the biological function of a plasma membrane transporter is going to be a lot different to the biological function of an inner mitochondrial membrane metal transporter. Localization is also very, very difficult to study right now. It requires GFP tags and complementation lines, so progress here is actually quite slow. And there's also regulation. This can be transcriptional regulation, this can be post-translational modification. For example, you could have, let's say, upregulation of a bunch of transporters in zinc deficient conditions. But what happens when you achieve that level of zinc that you need? Well, then you need to downregulate these transporting activities, which could be done, for example, by endocytosis. We're going to talk about the movement of zinc between different pools in plant roots. So I've made a diagram to try and explain some of the principles that are going on here. Now, remember, this is going to be massively simplified, but hopefully it's illustrative. So yeah, let's go. Just to key you in, their circles represent different pools of zinc. The arrows represent movement of zinc between different pools, and the labels, for those who care, are the Arabidopsis proteins that are responsible for each of these different types of movement. So from left to right, we start with environmental zinc. This is zinc in the soil, which can then be adsorbed onto cell wall components, for example, into the apoplastic flow of zinc. The apoplastic zinc can then be uptaken into the cytosol. Now, note how the cytosol here has two compartments. It has chelated zinc and also free zinc, with the free zinc being the minority. Now, from the cytosol, it can continue with some plastic flow towards the xylem, or it can be moved into vacuolar sequestration. I've only used one organelle here, but obviously, remember, plants have a whole bunch of different organelles. I've just tried to make it simple. Moving on to zinc deficiency now. So in zinc deficiency, zinc usage needs to be prioritized for developing seeds, flowers, and new leaves. And so it's controlled by a pair of transcription factors, BZIP1923. These regulate a bunch of transporters, but also nicotianamine synthetase. However, it also seems that there's a different shoot-specific zinc deficiency signal that can override a root-specific zinc deficiency signal. So just going from left to right, we have increased uptake from the extracellular environment due to upregulation of a bunch of transporters like ZIP2. There is buffering systems in the cytosol will have to adjust to reduce zinc complexation to maintain a cytosolic zinc concentration. There is reduced funneling of zinc towards the vacuole because there's no sequestration required really. And in fact, zinc transport out of the vacuole is preferred. And there's also increased zinc flow into the xylem through upregulation of HMA2 and 4, which transport zinc directly into the xylem. Now, I've just put roots on here, and I know it's very simple, but it's also missing all the above ground tissue. So what's going on there? Well, in addition to zinc in the roots, zinc stores in above ground tissues are also mobilized. And these include vacuoles in the shoots, but also trichomes. We've tackled what the flow between different zinc pools looks like in zinc deficiency. So now we're going to look at the opposite, zinc excess. So you have way too much zinc. Let's say you're trying to grow next to a factory that's pumping loads of zinc polluted material out. So here the activity of your transcription factor pair BZIP1923 is low. There is high amounts of environmental zinc trying to bind to the cell walls of a bunch of epidermal plant root cells. However, you want to reduce the flow of zinc from those apoplastic zinc stores into the cytosol. And so you downregulate these zinc transporters, whether that's transcriptionally, whether that's post-translationally, through endocytosis, which is quite common. You also want to increase the activity of transporters that then sequester this zinc. So this could be upregulation of your MTP proteins that cause increased vacuolar sequestration of zinc. There's also going to be upregulation of chelating proteins that will chelate the zinc within the cytosol, so like your metallothioneins, your phytochelatins, things such as that. And you also want to move your zinc to stores where it can be safely accumulated in large amounts. And so this could be in the vacuoles of roots, it could be in the vacuoles of the shoots, and that's why you also still have root to shoot transport of zinc in excess conditions. Okay, so we're done with the flow diagrams now, thankfully. This is my final slide. I wanted to talk about not just zinc, because zinc, although it's very important and very useful, exhibits a lot of properties that are similar, as we've discussed before, to other metals, and therefore the homeostasis of zinc is impacted by the homeostasis of iron and vice versa, and I want to talk a little bit about that here. So if we take a transporter that transports zinc and iron, not uncommon for a bunch of zips, and then you have balanced zinc and iron levels, then you're fine, you're perfect. However, let's say you have zinc deficiency. So the amount of zinc going through that transporter is now lower, and so the proportion of metals that go through that transporter that are iron increases. You then start to build up a bunch of iron levels in your cell. You then get iron excess through the Fenton reaction. You get increased reactive oxygen species, and you get oxidative damage. This is bad. Now, if we inverse it, where you have zinc excess, you end up with iron deficiency, which leads to chlorosis and a bunch of other problems. And so you see how this cross-metal homeostasis is very, very important to be perfectly balanced. And you need to use a bunch of different transporters with different specificities to try and optimize your levels of these different metals at the same time. So that's it for me. Thank you all for listening.